Okay, might as well get started. For those that do not know me, my name is Gus Kousoulas. I'm a professor of virology at the LSU School of Veterinary Medicine, and also direct the Division of Biotechnology and Molecular Medicine, to which a number of cores, infrastructure cores and service cores report to. Today, I will start the first of 3 lectures on protein production and purification. And these lectures are really intended to be at a very basal level, and the real reason is to bring everybody up to speed, especially in small campuses that may not have particular expertise or even the sophisticated, expensive equipment to perform protein production and purification experiments in analytical scale, perhaps pilot scale, but not necessarily at large scale. So we're going to focus on those 3 series on technologies that are readily available, focus on inexpensive or relatively inexpensive equipment that would be, whether it would be simple column chromatographies or even automated systems, as we're going to talk about next lecture, the Profinia system from Bio-Rad, which is only about $14,000, that perhaps a core lab in a small campus could purchase and have it used by a number of investigators, as would be also with a laboratory anywhere that could really purchase such a machine to be able to purify proteins and characterize them. So I'm going to focus on the first 2 lectures principally on the expression of proteins in bacteria, in E. coli, because this is a very robust system where protein can be expressed, large amounts of protein can be expressed. And then we'll follow with a purification lecture on the second lecture, which is, I believe, next Thursday, and I think I have a schedule on the slides. And the third one will be on expression of proteins in insect cell cultures using baculovirus expression vectors. And there are pros and cons on those systems, and I'll be discussing some of these here today. So as most of you probably know, is the molecular cell biology core of the IMBRE grant. The main purpose is to really assist the IMBRE laboratories in executing protein production purification experiments in their own laboratories without the need of sophisticated and expensive equipment, the presence of highly trained personnel. So to be able to do this, we'll do the training sessions, and we also will invite a select number of people to come in over a 2-day period to execute a specific protein production purification protocol that may need in their own laboratory. So these are the 3 lectures that we'll be giving. The first one today, the next one next week, Thursday, October 7th, and the following week we'll be doing, as I said, the baculovirus insect cell culture session. After we conclude all those 3, then there will be a questionnaire out, and we will recruit 2 or 3 labs based on their needs, people to come here, whether it will be students or faculty, to execute specific projects in our own core facility, which is now a brand new facility at Wilson Hall here on the main campus of LSU, where the idea is being that you'll be trained and be able to carry the protocol to your own laboratory and perhaps be an instrumental person or persons that will establish a similar capability, a core perhaps function in the small campus. So topics of discussion today in sequence, and I'll go through this very quickly. And mind you that this lecture today is really at a very basic level. So for some of you, maybe too basic. For some of you, perhaps maybe a little bit advanced. So part of the idea is to start really on very low level and try to build up from that. And as we have successive other lectures, we'll increase the difficulty, the types of vectors, the types of things that you could do with this. But I need to start from somewhere, and the best way to start is to start really at the very basal level, make sure that everybody understands what we're doing, and then build on that on next series of lectures that will have increasingly more advanced knowledge and capabilities. So I will start through going very quickly through the central dogma, molecular biology, going to cloning and vectors, discuss very simply, very quickly, differences between prokaryotic and eukaryotic expression systems. I will go into the type of inexpensive equipment that you could have in a core, in a laboratory to perform some of these experiments. Go through the anatomy, as I write here, of an expression vector. What are really the cis-acting elements, as we say in molecular biology, that dictate how a gene is regulated for expression? I'll briefly mention some cloning strategies. There are tons of manuals and possibilities out there to clone a gene in a very easy way, perhaps over minutes, into any vector that you like. Talk through a little bit about fusion proteins, introduce you to the principle of codon optimization, which is very important in expressing a particular protein in E. coli as well as in insect cells or yeast, and then go through some of the specifics of one system among the very many that are available, which is really the champion sumo protein expression system, which is sold as a kit by Invitrogen Corporation, as an example of a kit that could be purchased, and then the experiments could be performed in any laboratory. So the central dogma of molecular biology, everybody knows that. DNA goes to RNA, and then ultimately gets translated to protein. So DNA is the genetic information, and the actual phenotype is really described by the protein. This is really a world of proteins. We have approximately probably 25,000 proteins that make everything we are. And with the advent of new technologies in proteomics, we could simultaneously really look at all these 25,000 proteins, and the technologies increasingly become more sophisticated and more capable to look at the spectrum of these proteins. So, back here. In a more advanced way, obviously, DNA can replicate itself, and it can transcribe, but also RNA could be reverse transcribed into DNA. And of course, the RNA can make messenger RNA, can make ribosomal RNA, or transfer RNAs as well, ultimately be translated to protein. So based on this technology and with the advent of recombinant DNA, we can manipulate even RNAs, because RNAs could be copied into DNA. They really become double-stranded DNA, which is the easiest molecule to basically be able to genetically manipulate, to cut and paste, clone, propagate, replicate, and beyond that. We even have now systems to replicate RNA and make tons of RNA if we need to, so we can really circumvent some of the original problems we had of handling RNA in itself. Obviously, we cannot go backwards from protein to RNA, at least physically we cannot, but if we know the sequence, we could really synthetically make the RNA or DNA that encodes the particular protein. So this is the genetic code. Genetic code has a degeneracy that is multiple codons. Every 3 bases generate one specific amino acid. And ultimately, a gene is really what we call an open reading frame. It starts from an initiation codon, AUG, codes for methionine, and has a stop signal to stop really the reading of that protein in every triplet sequence. So you would have a specific RNA and methionine and a stop here. And the gene really is, the gene name in itself is really the open reading frame. Now, the gene can be regulated by cis-acting elements that sit upstream of the gene and downstream of the gene that really dictate how this gene will be expressed. But when we're really talking about a gene, we're actually relating to the open reading frame, that is the number of triplets that ultimately, without a stop codon, generate that particular protein. So, why do we use heterologous expression? Why do we're interested to really take any gene and express any protein? Obviously, that would be quite obvious, I think, to a lot of people, but these are some of the principal reasons. Proteins generally have a low natural abundance, so we need to express more of them in order to characterize them. there are proteins that could be predicted by reverse genetics. That is, we can know the sequence, but we do not know the protein. We have never seen it. So we can actually take that gene then and make the protein, generate an antibody, and go fish out where the protein is, or what does it do, or how does it behave in terms of its phenotype. We could also, with the advent of recombinant DNA, modify that protein at will by modifying the gene. We can change a single amino acid, we could delete domains, add domains, fuse proteins together. We could do everything that you wish in order to understand its function better. And ultimately, if it really, in instances where it pertains to an enzyme, we could try to modify the enzyme by amino acid changes to make the enzyme work even better than what is in nature. And you could do that through a lot of companies right now, where they look by random mutagenesis to really create molecules, proteins, that behave much better or have a much more specific role in a specific function than the originally intended through nature. So, gene cloning and vectors. Obviously, a lot of you know a lot about this, but I'll go very quickly through that. There are three basic features in all cloning vectors. Typically, these are vectors that permit the propagation of themselves in bacteria, yeast, or any other host system, like the yeast artificial chromosomes. Typically, they need a cloning site to insert a foreign DNA. The most versatile vectors contain a site that can be cut by many restriction enzymes. And as you'll see in some of the vectors, we have what is called multiple cloning site. That is a site that contains multiple restriction sites, so you can really choose, pick and choose which enzyme you want to actually insert a foreign DNA into it. And ultimately, we need a method for selecting for bacteria, either on yeast or yaks, containing the vector with foreign DNA. Usually, this is accomplished by a drug resistance gene, because when you have this plasmid into a bacterium, you really want to select against all other bacteria that don't have it, and you only want to bring out those bacteria that have it. So by having a selective advantage, a drug resistance gene encoded by this particular plasmid, you can actually select those bacteria for the ones that you want. Types of cloning vectors, and all of those are used extensively in many molecular biology laboratories, obviously plasmids. And the difference on this, as you'll notice, is really the ability to accommodate a increasingly larger size of a heterologous gene fragment. So a plasmid, it's an extra chromosomal circular DNA molecule, double-stranded DNA, that is autonomously replicating inside a bacterial cell. Typically, cloning limit is quite small, 10,000 to, 100 to 10,000, up to maybe 15,000 bases at max. The bigger the insert, the slower the replication of the plasmid, which means that typically a gene of 2, 3, 4,000 bases, depending on what we say the replicon, what kind of plasmid you use, may be quite efficient, but the bigger the fragment, the plasmid may not be able to replicate well. We also have bacteriophages that could be actually derivatives of bacteriophages, like the bacteriophage lambda, and those can accommodate much bigger DNA fragments, up to 20,000 bases. Cosmids, and these cosmids are, again, circular DNA double-stranded molecules that combine features of plasmids, so they have replication of origin from plasmids, as well as features from bacteriophages, and for that, you could have 35 up to 50,000 bases cloned into them. Bacteria artificial chromosomes, so BACs, these are based on a bacteria mini F plasmid, and they have a much higher cloning limit, 75 up to 300,000 bases. In fact, in our own laboratory, we have the entire herpes genome, which is approximately 130,000 base pairs, cloned into a single bacterial artificial chromosome plasmid. That plasmid can be replicated very efficiently in E. coli. DNA can be purified, extracted, and upon transfection of that plasmid into recipient cells, eukaryotic cells, African green monkey, or any other cells, you can recover infectious virus. So you can propagate the entire viral DNA into E. coli and rescue the virus in a eukaryotic cell. Yeast artificial chromosomes, or YACs. These are chromosomes that contain telomeres. These are the telomeres of fine eukaryotic chromosomes. An origin replication, a yeast centromere, and a selectable marker for identification and selection in yeast cells. And this is the host cell for the YACs. And those could go up to 1,000 kilobases, 1 megabase. And as you may know, one of the advances right now is that you really could reconstitute an entire genome, perhaps an entire bacterium, as he has it done by a couple different groups right now, reconstitute an entire bacterium by using YACs. So actually put together YACs and actually recreate all the chromosomes and really actually, in essence, create life by synthetically putting those YACs together. I'm going to really focus mainly today on E. coli expression systems using plasmids, but I think we're also going to go into, with the baculovirus system, we may focus on some other of these vectors in subsequent lectures, depending on what people are really interested in. So the basic steps to get a recombinant protein, obviously, is to really deal with the gene. We need the gene any way we can get it. Typically, in molecular biology labs right now, we could use polymerase chain reaction to amplify any gene target from a chromosomal target or from a viral purified DNA or RNA or any other natural substance that contains that nucleic acid. And by PCR, we only need a set of primers that bracket a specific gene. Then a polymerase chain reaction can amplify that 1,000 times, 10,000 times, 100,000 times, be able to give us sufficient amount of DNA to be able to go into the next steps. Also, we can now more and more efficiently synthesize these genes synthetically. In fact, for a number of the experiments that we do in our laboratory right now, we prefer to order the synthetic gene from GenScript than actually try to PCR it. Try to PCR it out from a substance. And there are some possibilities there because when you synthesize a gene, you could also change it. And as you'll see, I'll go a little bit more in detail later, you could also optimize its codons so that you can increase its translation. And this is the codon optimization algorithms that are used effectively. When you PCR a gene, you are actually PCRing the authentic gene. You're not really modifying it. And if you do modify it, you can only really readily modify the ends because of the primers, not necessarily what's in the middle, unless you do a more sophisticated PCR in steps and try to put those fragments together. So the gene is then made, either by PCR codon optimization, is inserted into a cloning vector, and now very many different ways to clone the vector, subcloned into a specific expression vector for expression purposes, transformed into bacteria, E. coli, and then after expression is performed and optimization of expression is performed, then we attempt to identify the recombinant protein, typically by using Western immunoblot if we do have a specific antibody. We could visualize it only by molecular weight electrophoretic mobility to make sure that this is approximately what we expect it to be, or any methods. We could hook it into a fluorescence tag and observe fluorescence if we have the right detector to detect fluorescence. Or we can even, if we have an antibody, we can do an immunoprecipitation from the bacteria E. coli extract to really bring out the protein and study it. Then if we're ready at that stage, and this is actually requires a number of optimization steps, we could go into a pilot or large-scale production. Obviously, the large-scale production can be done only in fermenters that would have 2 liters and up, 10 liters, up to 1,000 liters in biotech companies for actually producing a protein. The isolation and purification, this is a very tedious process that typically requires expertise because you have to go through multiple steps of really purifying the protein. But we hope to show you some methods where you could probably get 98%, 97% and above purity of analytically scaled proteins for your own use in the laboratory without necessarily having to go through a lot of pain in doing this purification. So, very factors to be considered. It's a complicated process, and it is an empirical process. There's nobody that could actually tell you, I'm going to teach you exactly how to do it, it's going to work 100%. It is really, a lot of it is trial and error, the right vector, the right conditions, the right optimization to get to the right conditions for that particular protein. But if you have experience, perhaps 70% of the time you could be outright right, and then the optimization will be only on the last 30%, especially if you have a very difficult protein based on its characteristics. So which expression vector to choose? Which host system? Obviously, we're talking now about E. coli, but it could be others. And then a very important factor is the properties of the proteins that you're trying to express. How big is the protein? How hydrophobic is it? What kind of amino acids are there? All of these are really very important for its ability to be translated effectively, whether it would be toxic or not in your bacterial host. That means it would actually kill the host before you can purify it, or whether it could be purified easily afterwards by a specific method, depending on its nature. So solubility, whether it has multiple domains, size, all of these are very important. It's also very important to know where, or predict, where it would be expressed in the host cell, or whether you want this protein to be secreted out of E. coli, or really expressed within the E. coli, so you could actually recover it easier. And then obviously how to identify the protein, isolate it, and which purification method you're going to use to really purify it. And there are a number of companies that sell tremendous number of reagents, how to clone, express, and purify proteins. I only picked this face page from Sigma here, just to show you that a lot of these companies are concerned about that and provide reagents. And you can see here that you can have if you wanted the characterization of a specific protein, you can go into prokaryotic cells, E. coli, bacteria, yeast cultures, you could purify it, and then you could do lyse it, extract it, test it in Western culture. You probably go into cell culture and so on. Ultimately, you have to analyze it, and during purification, you could use HPLC chromatography, affinity chromatography, and ultimately structural proteomics or quantitative proteomics to look at thousands of proteins at the same time. In fact, interestingly enough, there are now multi-plate format where you could express simultaneously thousands of proteins at the same time using bacteria expression systems in situ, in that well. And then if you had a screen, you could identify which of those wells is the one that you're interested in. If you had a specific antibody, or you had an enzymatic assay, or something like that. So you could do a random cloning and expression of everything that you're interested in and try to find out which one actually you want to come out with. So expression systems, as I said earlier, bacteria, not only E. coli, but you use Lactococcus lactis. There are many other bacteria that are very effective in producing large amounts. Some of the biotechnology companies use bacteria other than E. coli to express large amounts of protein. Yeast, as we mentioned earlier, insect cell cultures, and you can also express proteins in mammalian cells, and that will be probably the subject of a different session where we'll talk about expression of proteins in eukaryotic cells using shuttle vectors that could be propagated in E. coli in eukaryotic cells, as well as vector systems, where there would be recombinant adenovirus. Baculovirus is also used, believe it or not, to express proteins in eukaryotic cells, because baculo, although it's an insect virus, can enter into any eukaryotic cell, but it cannot replicate. But if you had the right cis-acting element in front of your gene, then you could express this gene in any cell. And there are also cell-free systems where you could, if you provide all the translational machinery in vitro, you could express this protein using either an E. coli extract or a eukaryotic cell extract like microsomal membranes. In fact, there are claims now that you could produce milligrams of a protein by a cell-free system. We have tried some of these in our lab. We're not very happy with them, but I suspect some other lab may be more capable than we are. They're quite expensive, but certainly the claims are there that you can use this extract, throw your gene in, and you will make protein. And you don't have to use bacteria, you don't have to transform anything, you don't have to do anything. You just provide all the necessary reagents to really make the protein in a test tube. So some proteins produced by genetic engineering, human insulin, growth hormones, plasminogen activator, bovine growth hormone, all these are now being produced by genetic engineering using either prokaryotic, E. coli, or eukaryotic systems, and they're produced at gram or kilogram amounts, whether it would be in Amgen, Genentech, or other of these companies. So everything that you use really has been produced through those systems. So let me show you then a typical prokaryotic expression vector and try to bring together what I mentioned earlier. So a prokaryotic expression vector, a plasmid would be used to express a protein in E. coli, is a double-stranded DNA replicon. By the term replicon, it means a double-stranded DNA circle that can really replicate independently or in conjunction with the host machinery, that is the E. coli machinery. So typically you will see an origin of replication, and most of the plasmids that we would use have a Coli 1 origin of replication, and then it will have additional elements that really relate to the expression. Mind you that the only thing you need to replicate this circle in E. coli is really the origin of replication. And then this specific origin of replication will allow the plasmid to replicate up to different number of copies. And in fact, if we're interested in expression, typically we're interested in a high copy number if we want to produce a non-toxic protein, but we may be interested in a low copy number if we're interested to produce a protein that may be perhaps more toxic to the cell. And then we have the area where we want to put our heterologous gene. Typically, that would be an area that has multiple restriction sites marked here by the polylinker. And then immediately upstream of this gene, we want to put cis-acting elements, and simply cis-acting elements are DNA segments that are recognized by the bacterial, transcriptional, and translational machinery. That's where basically RNA pol will bind and transcribe the RNA. But we may not want that RNA to be made all the time because it would be toxic. We wanted to make it at a specific time. So we have to develop systems, and people have developed systems, to regulate transcription and translation of that specific gene that would be inserted within the polylinker site. And obviously, cis-acting elements do not include only upstream cis-acting elements, as it is here the promoter operator region, ribosomal binding site, but also a downstream cis-acting elements, in this particular instance, a transcriptional termination sequence that allows for efficient stopping of transcription. And ultimately, we need the selectable genetic marker, typically antibiotic resistance, to select the bacteria that have this plasmid. So after we transform, that is, we inject the DNA into the E. coli, we select what specific antibiotic, kill everything that doesn't have it, and only retain the bacteria that have the particular plasmid. So, let's see. Why is it going backwards? So, in contrast, and I'm not going to focus on this, a eukaryotic expression vector is also called in many instances a shuttle vector, because it's a vector that can replicate in E. coli, make a lot of that, but also it can replicate in most instances also in eukaryotic cells. So typically you would have not only an origin replication for bacteria here, but in this particular instance you may have an origin replication if it replicated. This one actually doesn't have it for eukaryotic cells. But it will have also, your gene cassette will have ability to be transcribed and translated in eukaryotic cells. So the upstream and downstream signals for expression will be different. You'll be eukaryotic as opposed to prokaryotic. For instance, mRNA splicing, getting exons, and so on out of that. And of course, the promoter would be eukaryotic promoter, like the cytomegalovirus immediate early promoter, as opposed to a promoter, a bacterial promoter. So, and this is the expression for prokaryotic cells. We call this transformation, where the DNA would be put into bacterial cells. For eukaryotic cells, we call that transfection. That is, the plasmid is mixed maybe with lipofectamine, a lipid, to mediate entry into cells. or it could be electroporated, both in bacteria as well as eukaryotic cells, and then ultimately the expression will be occurring in the host cell. We could also, and this is more pertinent to eukaryotic cells, we could make permanent cell lines that are have a stable insertion of the heterologous gene cassette. So there's no more any plasmid replicating there, but the entire cell produces it. We can also do that in bacteria cells. In fact, some of the host cells that we're using for the expression systems are modified at the chromosomal level to express, for instance, the T7 polymerase. So we can make a bacterium that has a specific gene into its chromosome, but typically those are really modified only as hosts, not necessarily for expression of proteins. And this is, well, I'll go backwards again. So now, what a typical lab, very quickly I will go through this because it's very basic. What would you need to really do expression? What would be the types of needs you would have to go into an expression experiment? Obviously, you need to have a target, a specific gene of interest. You can synthesize the gene, you could amplify it, somebody will give it to you, and you can work on that. You need to have an expression vector. You'll choose a vector. And today we'll be discussing some of the vectors for bacterial expression. Your host cells, and you have the ability to grow them and characterize them. And I'll go through those specifics. So the host systems, obviously E. coli. Why? Because it's very cheap when it really works. You can make tons of proteins. Easy cloning and genetic manipulation, inexpensive to culture. It's very rapid to grow and very fast expression. The equipment are needed, could be categorized under these categories. Basic equipment that any laboratory probably will have. Cloning, equipment that would facilitate cloning. Expression, specific equipment, and ultimately ability to purify a protein using specific equipment. So, basic equipment, I think you can think of it what it would be. Anything that could manipulate DNA on your bench. You need ultra pure water system. A lot of problems if you don't have good water because you have contamination and proteases and a lot of bacteria that could contaminate this. Obviously, that also pertains to using the right pipetters and sterilized microcentrifuges, tabletop centrifuges, preparative centrifuges, not necessarily a high ultra centrifuge, but preparative that I know a lot of the small campuses do have. Benchtop shakers to really grow your bacteria, obviously autoclaves to really sterilize your media and everything else you'll be using, and buffers, pH meters, stirrers, and stuff like that. Now, if you will clone your own expression vectors, obviously you need to be knowledgeable in basic separation technologies using for molecular biology, agarose, acrylamide gel apparatus, and power supplies, heat packs, water baths, ice buckets, DNA purification kits, and this time you could buy a lot of these from different companies such as Kiogen, vacuum pumps, vacuum supplies to be able to evaporate some samples and concentrate them, obviously incubators and shaker incubators, tubes and flasks. Specialized equipment for cloning that are very effective, PCR machines, spectrophotometers to be able to really determine how clear, how good your DNA is by getting OD ratios. Access to DNA sequencing will help to make sure that you have the right segment cloned correctly as you did. But all those could be also accessed. We do have a sequencing facility at the LSU Vet School. You could send your DNA, could be sequenced. There are also a lot of companies out there that you could mail them in an envelope, your DNA sample, and 2 or 3 days later, you're going to get a sequence back. So for expression, incubators, a fermenter will be optional. There are small tabletop fermenters that could do 1/2 liter up to 2 liters if you really were serious about expressing proteins. And you could do a small-scale power optimization if you had other more sophisticated equipment. There are a number of those at Pennington Biomedical Research Center here, as well here in this building, to be able to do 96 or 24 well plate capability for expressing up to 96 bacteria clones simultaneously. Now, if we look really very quickly at expression systems characteristics, and I'm going to go through some of these a little bit more in a while, you could see that there are really major differences between E. coli, yeast, insect, and mammalian systems. The more important is ability to really replicate those very quickly. You could see within 30 minutes, doubling time of E. coli, inexpensive. Here we have cost of growth being very low. And the amount of protein could be produced is quite high, but there are also a lot of negatives, and I'm going to go and explain some of these a little bit later. But typically, you're expressing a protein, and if you're really wondering or you're interested in a eukaryotic protein that could be post-translationally modified, as it is glycosylated, phosphorylated, otherwise processed, myristylated, then bacteria cannot do this. So you would have to consider a eukaryotic system if you're interested in those post-translational modifications listed here: acetylation, phosphorylation, glycosylation, and so on. But if you have a protein that you're interested, that it retains its phenotype, that is its characteristics in bacterial cells, then the cost is very low and it's very quick. So you can produce this protein. A number of the proteins that have been crystallized, the X-ray structure has been derived, actually have been produced in bacteria, because a lot of protein is required to make crystals, and bacterial systems are very efficient in doing that. So let's go now into anatomy of an expression vector, critical vector elements. I mentioned already the origin of replication, which will dictate whether it will be a low copy, a high copy number, antibiotic resistance. In terms of the cis-acting elements that sit in front of your gene, strong promoters, ribosomal binding site, obviously a multi-cloning site, a vector that has multiple restriction enzymes that you can clone your gene any way you like, and then you could pick it out a way you like. That would be very important. And you can have also various cloning vectors that have already code for tags. And tags are small amino acid segments that already have been targeted by specific monoclonal antibodies or monospecific antibodies. So if you tag a protein with this specific amino acid domain, then you can use that tag to really characterize the protein without necessarily having an antibody against the protein itself. And if you do have a tag, or you have some other carrier that you're expressing the protein with, then you need a method for cleaving off this unwanted portion. And most of these vectors, and I'll show you some of these examples, contain protease sites between your tag or your carrier and the protein of interest. So after you purify it, you could cleave it off and then only isolate the protein that you're interested in. So in terms of the gene expression sequence of events, so we start with DNA. Ideally, a high gene copy number would be good, especially for smaller proteins or non-toxic proteins. Promoter activity, we're concerned with how strong is that promoter activity, how effectively polymerase can initiate transcription. We're concerned about mRNA stability. Is that mRNA stable enough? And that's actually an art in itself, because you can go into secondary structure and other considerations. A ribosomal binding site, because we want the ribosomes to really clip onto that initiation site and start translating protein. We're concerned with codon usage, because it turns out a bacterium uses different codons than a eukaryotic cell, prefers better triplets, different triplets than eukaryotic cells. And by changing your codon usage to E. coli-like codon usage, then automatically you get better translatability. The same thing is true for a bacterial gene expressed in eukaryotic cells, or for a eukaryotic or prokaryotic gene expressed in insect cells. A codon optimization, that is a usage optimization for codons in a particular host, is the first step for increasing translatability of a gene. Now, in terms of the protein, we'll be concerned with protein stability. That is the protein stable after it's expressed, or will be degraded? Folding, where they will be forming inclusion bodies, and then we cannot retrieve it very easily in bacteria. And of course, whether we needed to have post-translational modification that will dictate whether we'll choose a different type of expression system. This is now a vector, a plasmid vector for bacterial expression. As you can see here, we have a multiple cloning site. We can knock out rare codons that maybe we do not want in E. coli. That could be done by codon optimization. It has a start and a stop. And in this particular vector, we also have fusion tags that are in frame to the protein, and those could be added at the amino terminus over here, also at the C terminus. In certain instances, if we have a double tag protein that has a one tag in the amino terminus and another one in the carboxy terminus. Ahead of this gene cassette, there will be a ribosomal binding site and a promoter regulatory element that will control the expression of that gene in the bacteria at the time that we're interested to really express it, because we don't want to express the protein very early in the replication cycle of E. coli, because then the E. coli will not grow and they will die. We want it to be really at log phase before we throw an inducer to make the protein, because that will maximize then the amount of protein that's expressed. And I will go into the specifics of this a little bit more as we go through this. So this is now a gene cassette as it stands in most of these vectors. We have the gene of interest, open reading frame, encoding a number of different initiation codon, ATG, and a stop codon over here. Upstream, we have the controlling region, promoter, operator, and ribosomal binding site, and downstream, the terminator over here. The operator is the area where the RNA pole will bind and transcribe, and typically in every plasmid or host system, we provide a repressor, which sees the natural state of the operator is ready to be stopped. So there will be no transcription unless an inducer, a molecule, come in and de-repress that particular operator to allow the RNA pole to initiate transcription and translate the protein through the promoter binding site. Replication of origin, antibiotic resistance. So let's look very specifically at some of these sites. These cis-acting elements, again, they're really DNA segments, small DNA segments that sit upstream of a gene, and there are consensus sequences for really binding either the ribosome or binding RNA pole 2. So you have ribosomal binding site, a spacer sequence. In most instances, it's quite important to have that ahead or between your ribosomal binding site and the start, which is the initiation codon over here. And because we know these promoters, we can really synthesize those promoters. Synthetically, we make not only the gene, but we can make a gene having the upstream promoter elements that we need. So if we're knowledgeable what these upstream promoter elements are, we can really synthesize a gene and clone it into another vector that we're interested in, and then we impose the regulation of that gene under the prescribed upstream sequences. Now, in terms of start codons, in terms of codon usage, E. coli uses 77% of AUG, which is the ATG, and with less frequency, 14%. So what do you think would be the codon that you would like to use in E. coli mostly? AUG, because that's the most frequent codon that's actually used for initiation. And stop codons, all those are used quite effectively. So I already mentioned that this promoter operator is really the area where control of transcription is initiated. In most instances, we do not control translation, but really transcription. There are very few systems that I'm not going to go into that may be able to control also translation. The idea is never to make the RNA, or make the RNA when you're really interested to make it. And then in this slide, you can see those a little bit more analytically. So the promoter consensus sequences, there are spacer requirements ahead at minus 35, and typically those are found in most genes at minus 35, minus 10. Transcription start site +1. This is the +1, and this is related to this, this particular site here. The ribosomal binding sites, the Saint-Algano sequence, translation initiation, and then ultimately you have transcription start and the translation downstream from the transcription start site by recognizing the ribosomal binding site. So promoters, there are 2,500 known promoters in the entire E. coli K12 genome. You could pick any of these promoters and make your own promoter operator region to respond to a particular element. And in fact, if you look at all, whether it be Invitrogen, Novogen, you will see some fancy names, PTAC, PLAC, PBAD, and all of those are really different gene elements, promoters, that have been combined with their ability to regulate that operator promoter region to really respond to an exogenous stimulus, whether that would be IPTG, which is an analog of lactose, or it would be arabinose, or it could be some other system. So it really just pertains to regulation of that. We can also use promoters from bacteriophages, and one of the best promoters, very strong promoter, and I will explain that, is really the T7 bacteriophage promoter. That promoter could be inserted ahead of a gene of interest to be expressed in E. coli, but it would require the T7 polymerase from bacteriophages. So what people do, as you'll see, is then create a bacterium that constitutively express the bacteriophage T7 from its chromosome. So then that gene can be recognized only by the T7 polymerase. And if you control how the T7 polymerase is expressed, then you can control whether that gene cassette is actually transcribed and translated. And I'm not going to go into the details of this, but this is bacterial genetics 101, which is really the lactose operand regulation. We have the promoter, the catabolite activating protein, and under different conditions, if you have a IPTG, which is an analog of lactose, you could de-repress a gene and cause massive transcription. When the operator is really de-repressed, then the promoter is active, polymerase will bind to the polymerase region. It won't be stopped by anything blocking at the operator region and be able to transcribe through the lactose, beta-galactosidase, and the genes of the operand. Now, types of expression vectors, very quickly, transcriptional and translational fusions. And you'll see those in different plasmids. If you go to any of the manuals or websites from these companies, you could quickly recognize whether it's a translational or a transcriptional fusion. In a transcriptional fusion, you insert a signal here with its own translational signals. That is, you make a gene that's not only the open reading frame, but it has the Shine-Dalgano sequence and upstream sequences that you would like to put in there. So then it becomes a transcriptional vector because you're actually providing not only the gene, but the elements upstream, perhaps even downstream, that you'd like. And you allow only the promoter region to be really part of your plasmid vector. So in a translational fusion, the promoter and all the translational signals are already provided by your plasmid. So the only thing you need to do is really insert your open reading frame, starting from the ATG to your stop codon. And this is the easiest because most of the companies already created a strong upstream regulatory region that has both promoter as well as translational control signals, that is Shine-Dalgano sequence. The ribosomal bind site and start codons may also be provided. In certain plasmids, a leader sequence, where there would be a signal sequence that leads to secretion of a protein, may also be provided. So you're only cloning part of your protein past your initiation codon, and you accept the fact that there would be another amino acid sequence that is expressed in front of your protein. And if it's a signal sequence which is cleavable, it could be cleaved. Therefore, you're not going to have it on the resultant plasmid. Now, this is from Invitrogen, a sophisticated vector, and you can see now all the elements that I talked about here. You have your gene of interest, and that gene is tagged on the amino terminus. Actually, we have initiation codon, and after that, you have a 6-histidine tag, and 6 histidines in tandem are used effectively for purification purposes. You have now all the upstream promoter elements, your ribosomal binding site, and your multiple cloning sites actually used here, where the gene is actually inserted. And in between your tags and your gene of interest, you have the protease, and that protease then will be used to cleave off the unwanted part, your histidine tag, and release only the purified protein. And I'll go into this a little bit more detail in a little while. Now the promoters, as you see here, we have the LAC operator. This is the P-tag promoter, and those are inducible by IPTG. That is, IPTG will displace the repressor from the operator region and allow transcription through this gene cassette, meaning that if the bacteria are grown to log phase, you do then put in IPTG and you get massive transcription and translation. And typically you want to catch the bacterium at the right growth stage, and you want to put the right amount of IPTG, because if it's too much, it may not do good. If it's too little, it may not do good. And you have to do optimization to actually get the best expression. Now, cloning. I'll briefly mention how do we clone things. And I see some people that come up to me and they still use archaic methods for cloning genes. It turns out there are very sophisticated methods and very fast methods to clone a gene. When I was a graduate student and started cloning in the early 80s, we were putting genes together and using ligase and waited 24 to 48 hours for the ligase reaction to occur so we can put that gene together into a plasmid. Now you could do this gene cloning really in minutes. You could throw a segment in and it will ligate into your vector and you're ready to transform into bacteria within minutes. So one of them is restriction enzymes. A plethora of them, you cut them, and then if you have a gene that has these sites at the ends, you can clone it directly. You could also use a PCR product, add restriction sites to it, and then clone and ultimately ligate into your plasmid vector. Newer systems, they're already plasmids that have a T overhang, and that capitalizes on the fact that any PCR product creates an A adenine overhang, and that A then pairs with a T, and upon ligation, you could actually clone the gene very quickly. You could also have a topo kit or topo mechanism using topoisomerase. This molecule, the vector is provided open with a topoisomerase molecule from vaccinia viruses covalently linked to its ends, and that topoisomerase has also ability to ligate genes together. So any gene you put in there automatically will be cloned based on the right sites directly into the plasmid vector, and Invitrogen provides explanation for that. So all the topo kits that you see from Invitrogen are really based on topoisomerase-based cloning. I'm going very quickly through this. Could devote lectures perhaps on those how it's done. A very powerful system for propagating a clone to thousands of clones at the same time is the gateway system. And this gateway system allows you to clone into an entry vector, and then if you know what you want to do with this, you could very rapidly, within minutes, literally, clone this original clone into hundreds or thousands of different vectors by a simple recombination event occurring by two specific sites and a mix of enzymes that cuts, recombines, and ligates and transfers your plasmid of interest, your gene of interest, into any other vector. So let's say if you had 100 different expression vectors and you didn't know which one really could do the job, you could go into an entry vector and then very quickly get 100 different clones in all the vectors, and then screen which of those vectors will do the job. And that's what's done typically in companies using the gateway system. They also do that in 5,000 or 10,000 clones to really try to get what they're interested in. I mentioned copy numbers, and some of the vectors that you see here, they have 500 or more copies. Some of them could have low copy. Typically, if you have a toxic protein, you expect it to be toxic. You want a low copy plasmid because you want to express it, but you don't want to express it very quickly and very rapidly. Otherwise, it will kill the host. You will never be able to isolate the protein. So the disadvantage is you're not going to produce a lot of protein, but at least you can get some using a low copy number. And expression strains are being E. coli strains that have been genetically modified to optimize every single aspect of what I just talked about. better promoter function, better expression of RNA polymerase, better stability of RNA, okay? Or maybe have been inserted T7 polymerase from bacteriophages so they can express constitutively T7, so you can use a T7 promoter in your gene cassette. And all of these, the most famous one is obviously BL21 and its derivatives. You can see this one here is deficient in RNA-C, so improves stability of mRNA transcripts and increased protein expression yields. This one here carries the T7 polymerase under a PLAC promoter, so it's also regulated by the same IPTG mechanism that I introduced earlier. So not only you regulate your gene expression, but you also regulate T7 expression. And if your T7 is the promoter you're using, then you need T7 polymerase in order to express the gene. So I'm not going to elaborate on this. And this is another list of many others that have different mutations. And by picking the right strain for your experiment, you could have maximum protein expression by optimizing mRNA stability, transcription, translation, and stability of the protein itself, because certain strains have been selected for their ability, or at least to not degrade certain proteins. Now, I mentioned fusion proteins already. You can use small tags. You can use larger tags that are fluorescent, like green fluorescence protein. You can redirect proteins into secretion in E. coli by using the secretory mechanisms of bacterial proteins. You could display a protein at the top of a phase, like a bacteriophage. In fact, one of the interesting things of find monospecific antibodies that react against a specific domain is to really take a protein, let's say, with 1,000 amino acids, and subclone in all permutations every segment of it, and have it expressed on the surface of the bacteriophage. And then select with an antibody which bacteriophage is actually expressing the immunodominant epitope, and identify that by picking the epitope and sequencing the gene after that. So the fusion can happen for purification or identification purposes. You could have your protein target and epitope target next to it. You could have it downstream, upstream. You could also use soluble proteins to improve the solubility and to facilitate purification. For instance, the maltose binding protein, which is a very large protein, could be used to increase the solubility of a protein that may be forming inclusion in E. coli, and you'll be able then to purify it much easier by using a larger tag than a smaller tag. And again, I mentioned signal peptides or other proteins to promote secretion. Now, there are many expression purification systems available, and if you go into Invitrogen website, you're going to see a lot of those. And you can have FLAG and polyhes, glutathione transferase. I mentioned maltose binding protein. You could have those in the amino terminus of the protein. You could have them in the carboxyl terminus of the protein. And those are done for solubility purposes, for stability purposes, sometimes in many instances for purification purposes. And we're going to demonstrate next week, use the PGX vector, how does glutathione transferase is used as a fusion protein, as a fusion tag to facilitate immunoaffinity purification using an automated system. This is another vector from Novogen here. The same thing. You can see now the T7 polymerase sitting promoter sitting upstream of the lac operator region, ribosomal binding site ATG. This is your his tag, and this is the site where you're cloning using a topoisomerase mediated recombination over here. And this is a different one as a V5 epitope, a small epitope tag recognized by monoclonal antibody to V5, sitting downstream, as well as 6 his tag in tandem. So you have more than one tag in tandem, 1, 2, or 3, and see which one works the best to actually use this. And you can see here the purification. This is a soluble fraction of E. coli, and in this particular instance, they're using a additional protein to stabilize it. And I'm not going to go into specifics here, but you could do multiple tricks and optimizations to try to get the protein of interest. This is another vector here. that uses the topo cloning capability. This is a PCR product accepting A complementary sequences into a T site. The vector is provided, ready to clone into it. You put this into that and ligate, and then either through the topo or direct ligation, you get that in here. And there's also T7 promoter expressed in here. And you can see in a Kumasi blue stain, large amounts of the protein before even you actually purify it. So by using a Kumasi blue stain and a high efficient expression vector, you can create milligrams of protein literally in less than 100 to 200 mils of bacteria culture. Small tags, they can improve expression, and I will discuss very quickly, or I'll point you, if we don't have time, to a kit by Invitrogen that uses the SUMO protein as a fusion. It can improve solubility. You can target specific compartments, whether it will be cytoplasmic or secretable. You can improve detection by having a monoclonal antibody or a fluorescent tag. You can improve purification by using ion exchange, affinity chromatography, and other methods. For large peptides, maltose binding protein, HIT, cellulose binding domain, glutathione transferase, those have advantages and disadvantages. The obvious disadvantage is now if your protein is already 1,000 amino acids and you're cloning in front of it another 400 amino acids, you have a much bigger protein, so therefore the amount of production will go down because these are rather big protein. The advantage would be that maybe now the protein is more soluble, and now you got a big tag that you could really use to purify the protein from. Proteases that could be put between your tags, and a number of them is just a very limited list here. Some of them very effective, like the tobacco aging mosaic virus, which is a plant protease. That sequence is put because TEV protease is not found in eukaryotes. So you could actually use that and modify the protein without having worry that you may be contaminating any other system that you use subsequently to that. Now, I'll go very quickly to introduce the concept of codon usage, and forgive me if I'm going fast because already I'm past an hour, and I wanted to allow a little bit of time for questions. So codon usage, you recognize that there are 64 codons. That's actually graded to amino acid coding sequence. There are many synonymous codons that different due to the degeneracy of the code. There are different codons that generate the same amino acid. But are all these synonymous codons translated with equal efficiency in a particular host? The answer is no. Some codons are translated more efficiently than others due to specific transfer RNA abundances. That is, there are certain tRNAs that are not available in a specific host. So if you have the wrong triplet, it won't be translated. It will be a stop. So the protein will stop there and it will be truncated and degraded. And that's why we're interested in having codon optimization, because we want to make sure that certain untranslatable codons or very low translatable codons are not there because basically they're going to kill translation of that protein. And we talk about codon adaptivity index over here, frequency of codons found in different expressed genes, and all these really statistical derivations to find out which codons are really more frequently in what host. And those tables are made. And I wanted to demonstrate this, but perhaps, John, it will be complicated to go into this website. But you're going to have these PowerPoints, and you're going to see this website. There are really free websites where you can put your gene of interest, and then you could optimize it to use statistically the codon usage of a bacterial host. So you could take a eukaryotic gene and codon optimize it for bacteria expression, or you could take a bacterial gene and codon optimize it for insect cell culture expression. That is, avoid then those infrequent or random tRNAs, AUG codons that will not be translated, that could be a problem in terms of translation. So again, in terms of advantage of the E. coli, we talked about this, how cheap and good knowledge of the host, genetics. We can manipulate the host to provide genetic elements. We can manipulate the plasmids, and we have very fast growth. But there are also disadvantages. We have limitation for expression of eukaryotic proteins, tRNA, so we need codon optimization. This is the first thing that you would ever do to really get efficient expression. And obviously, downstream translational, post-translational problems. If you really want it phosphorylated, you may express a protein that may not behave. The phenotype of the protein may not be directly assignable to what you see in your eukaryotic cells. For instance, the phosphorylated version is the one that has activity. The unphosphorylated version that's expressed in bacteria may have no activity, or may not fold correctly, in which case you're really not studying the protein. There are also a number of other problems relating to toxicity. The inability in bacteria to form disulfide bonds. So if you're actually looking to really create a dimer of a protein that uses sulfohydryl bonds, you may not be able to do that. Folding ability, and we mentioned already the codon uses that could really be overcome by codon optimization. One of the biggest problems in expression in bacteria is protein folding. If you overexpress any protein in E. coli, there's a high chance that it will form what is you see here, what is an inclusion body, a high dense aggregate of protein that's really insoluble unless you use 5 molar urea and dissolve it and then try to reconstitute it. So, and this is the problem here, where you can see total expression is over here. The insoluble part, all of the protein under Kumasi blue, is in the insoluble part. The soluble part, which is really the supernatant after precipitation, has very little of the protein. And there are now ways to even take the insoluble part, solubilize it, and then reconstitute it, and still retain the protein. And we'll probably show some of this next time. Now, I want to really go very quickly and tell you a little bit about this Invitogen Champion PD SUMO protein expression system. And this is really a kit that Invitogen sells for high-level expression and enhanced solubility of recombinant proteins in E. coli and cleavage of the native protein. So this particular plasmid and expression system attempts to increase translation, but also provide a protein that will enhance solubility and protect the protein from degradation, as well as having ability to really cleave the protein of interest out of it and then purify it. And the SUMO expression system consists of the vector itself that has at the very amino terminus a SUMO fusion partner. This SUMO is an 11 kilodalton protein that has been shown to increase expression level and solubility of fused proteins, also a substrate for the SUMO or ILP protease. So this SUMO protease is the key to the system. Rather than cutting at the recognition sequence, it recognizes the entire SUMO complex. This recognition is very specific and eliminates non-specific cleavage, a common problem with other proteases. So this one can be used for multiple expression experiments, as you see here. But typically, it leads to a soluble protein, high-level expression, with a gentle cleavage that will not really affect the protein itself and lead into native protein purification. And this is the Invitogen website. It has a wealth of information, not only for what you could buy, but also manuals for the entire experiment. And you go through the manual, you could download the PDF of a manual. You can see cell-free expression here, prokaryotic expression, yeast expression, insect expression. And those manuals contain step-by-step instructions of how to use the kits that are rather expensive, but if you get to use them once, you can retain some of these vectors, and then you could propagate your plasmids and express them without necessarily going back and buying additional vectors. Now let me go out of here quickly, and then go here. Oops. Where is it? Can I close this, Mark?
That's good.
Do you see it? Go ahead and open.
What are you looking for?
The PDFs?
There's right there.
Yeah, the Adobe Reader.
Click on that. There's 3 of them there.
All right. Let's see. Let me just show you this first. This is the vector that we're going to discuss next week. This is a vector from GenScript, and it has a T7 promoter and the glutathione transferase. And this is a fusion, histag, multiple histidines, GST, and a cleavage site urokinase. We will demonstrate the production or a strategy to produce monospecific antibodies against a segment of a protein that has been produced and expressed and purified by affinity chromatography using this vector here from GenScript. Now, GenScript is also a company that will synthesize any gene of interest. You submit your gene, they have a codon optimization protocol, and they will send you then an estimate of what it will cost. You select your enzymes upstream and downstream, and they will optimize it for bacterial expression or insect cell expression. Typically, any gene in 1,000 bases will cost probably $300. So it's not really as expensive as it used to be. The bigger the gene, the longer the gene, the more expensive it will be. And let's go here. This is now the manual for the Invitrogen champion system. And I will go, if I can, very quickly to show you the vector. It has step-by-step instructions. It tells you all the reagents. I'm not sure exactly how expensive it is. But it's probably the $400 range. And I'm just going to go into and explains all the things I was telling you earlier. It shows the multiple cloning site, how you clone into it. It should be somewhere here at the end. Even goes into how to analyze the proteins, the purification, and then it's a long manual. There it is. And this is the Suno protein. You can see again that you have your operator promoter region. The promoter is the T7 promoter. You use this bacterium in the context of BL21 cells that constitute actually inducibly express T7 polymerase. And you do have your histag and sumo protein encoded on the amino terminus of the protein itself. And this then leads to better solubility, stability, and expression. And you can use the sumo then protease to cleave this part off and release the histidine sumo part away from the protein of interest. I was going to go through some of the basic elements of this, how it's done, and point you to some of the specific notes in this manual, but I think you could do that on your own. And if you have any questions, you could email me, ask me how it's used. And as we go through the next 2 lectures, you'll see after the 2 lectures are done, we will advertise a selection of possible projects. So whether it's a graduate student or a faculty member, any of the PUI campuses, as well as here at LSU, may be able to accommodate a specific experiment. Because what we really want to do is, although we do have a core facility that does this, and it does that in conjunction also with a private company, really what we want is individual labs, individual students and faculty to be able to use this on their own, in their own laboratories for their own specific experiment, with us really being perhaps the more knowledgeable, or at least a partner to discuss with you what we have found and what may be a better way to do it, so you could actually be able to do it more effectively in your own setting. All right, thanks very much for your attention, and I'll be happy to answer any questions.
You might want to go through each site and ask them for questions. Ask each one.
Well, I mean, you know, this is basic information. I really wanted to bring into focus today what are we really talking about. And there's a lot of information, and some of you may be already very knowledgeable in this, and some of you may not. Because this lecture, this session is videotaped on the LBRN website, you could really go back and review it at your own leisure and see the slides. And if you do have specific questions that may better formatted in your own mind, you could email them to me, or we could even set up a video conference to discuss it. Or you may be able to come with these questions on the next session, where we're going to go more into the nuts and bolts of how do you really deal with a protein, a specific protein. We'll probably be dealing with a West Nile protein that we have already cloned, purified to try to generate an antibody, or a armadillo protein that one of our colleagues is interested in. So anybody has any question for me? I will be happy to answer it. Don't be afraid to ask. There's no dumb question.
ULM, do you have any questions?
Actually, we are stuck up on, we are trying to extract protein from BL21 strain.
Right.
But we cannot right now afford to use French press.
Okay.
So we would like to know if there are any other equipments which we can handle other than the sonicator.
So you in essence, you cannot solubilize the bacteria.
Yeah, we are afraid that if we use the sonicator, we may, like the cell lysis, it may interfere between the actual pure protein.
So you're trying to basically break the bacteria without using any solubilization, any detergents.
Yeah, we would like to know also that if there are any buffers that can break this bacteria.
Well, people have used mechanical methods like the French press to do similar experiments, but there are a lot of very mild detergents that could be used to facilitate solubilization without necessarily destroying the nativity of a protein. So Pierce has a number, Pierce Chemicals has a number of detergents that are specifically made for preserving native conformation. So if you look at the Pierce website and look at some of the detergent kits, in fact, there is a kit where it provides multiple detergents, and you can test which one will be more optimal for what you need to do. And after you see which one is the best, that you proceed with that.
Sure.
So I would check Pierce website.
Okay.
That would be the first one that comes to mind. I'm sure there are others, and there are some articles on that. So if I identify a couple of the articles, email me, and then I could try to reply to you and send you some of the specific articles of how this is done.
Thanks. Sure.
Yeah.
Any questions from Louisiana Tech?
Any of you interested in expressing a protein in bacteria or making a monospecific antibody in collaboration with us at the vet school? Are you thinking, do you need to express a protein or to make an antibody or any of that? Or any of your colleagues you think may be interested in that? Well, let us know.
So far, the antibody we've been using is mainly commercially available. And we haven't encountered any we need to customize and made in the facility, but we'll keep that in mind.
Yeah, well, obviously there are a lot of antibodies are available. A company like Abcam has many antibodies. Typically, some of these antibodies are very expensive, especially if you use them repetitively. If you want to use it only once or twice and you can spend $300 for some monoclonal antibody, then it's quite useful. But if you want a lot of antibody because you want to do affinity chromatography or for some other reason, then it may be cost effective to really try to make your own monospecific antibody for that usage. But you're right, there are a lot of antibodies available. But if you had to generate, but there are also a lot of proteins that do not have specific antibodies, especially if you're working, in armadillo or turtle or some other host. You may not be able to find monospecific antibodies that are specific for that, so you may have to generate them. But antibody production is one application of protein expression because you make it the antigen. You can create a vaccine, obviously. You can use a protein for diagnostic purposes. You can use a protein to crystallize and get its X-ray structure. So proteins are fundamental. Really, the true phenotype, how we behave, how we look, is because of proteins. So expressing a protein can find multiple uses besides just making antibodies.
On that note, we're actually probably interested in looking at the protein crystal structure. And in that case, how much protein amount do you think that's needed for that technology?
Yeah, we do have here in the Department of Biological Sciences, we have a group of people that express proteins in bacteria and have gotten X-ray structures. Marcia Newcomer would be one of them. And typically you need milligrams to gram amounts of protein because the crystallization process is quite inefficient, and until you arrive at the crystal, you need a very large amount of protein. You cannot do it with micrograms or nanograms of protein, so you would need milligrams, typically 100 milligrams, 50 to 100 milligrams above. I don't know if you know more about that or anybody else. But you need large amounts of protein, and they have to be relatively very pure, at the level of 99% and better, which then means that you have to have tandem protocols of not only affinity chromatography or other types of chromatography, but perhaps even cleanup, high pressure liquid chromatography or other methods to arrive at the very clean protein. But we do have capabilities here. There are a couple of investigators that could come and speak specifically to this subject. They could give an example how proteins expressed, purified, and crystallized. So we could, if there's interest from everybody from the PUI campuses, we could invite one of those investigators here to give you the specifics, not only on the protein production purification, but ultimately the intermediate steps of how do you crystallize, how do you get the crystal before you get the crystal structure.
Thanks.
Anyone else at Louisiana Tech with a question? Okay, that leaves you, Tara, LSU's report, if you have any questions for Dr. Kousoulas.
Okay, can you hear me now? I had to turn my
Yep.
audio back on.
Yep.
I just wanted to clarify, you said that next week we'll get the application of this technology?
Right. We're going to talk about a specific example of how to, we have expressed the central protein. We'll demonstrate the machinery, how it's done in a small automated affinity chromatography system that perhaps could be also installed in any laboratory on any small campus very easily. Does not require ability to be a chemist and understand chromatography. I call it a dummy system because you push a button and it does everything from the beginning till the end through a kit. And we're not really chemists, and we have worked on this for a couple of years now, and we think we understand it. So we hope to transfer this knowledge to anybody else that may be interested.
Do you know how easy it would be to obtain travel funds to visit the campus and actually have hands-on training sessions?
Well, what I hope is, because this system right now, Mark, you may correct me on this, is about $14,000. It may be even less now. It's easily within the budget of the campuses. So I suspect some of the funds among investigators leveraged with some institutional funds or overhead recovery could be used to purchase a piece of equipment like that. And then if you had a sister equipment like this, you could really identify the protocols and use it with us. Obviously, you're also welcome to come here if you wanted to and do an experiment. And we will try to invite 2 or 3 people at a time to come here over a 2-day period to perform specific experiments. But the ultimate goal is not for us to do it or have it done here, but perhaps have ability elsewhere. And the more people do similar techniques and they're part of this forum, then we're going to learn more. But because different people will be using different vectors and different conditions, and we hope to post the experiences of everybody on our LBRN website. Perhaps have a blog where we can change opinions and help each other for our own specific experiments.
Gus, I think the question was, is there going to be any travel funds available for them?
What was it, travel
funds to come do hands-on training here?
Okay. I'm sorry.
Is that right, Tara? Was that your
Yes.
question? Travel?
Oh yeah,
Yes.
I didn't hear the travel. I heard the how expensive was the equipment. So apparently, Mark, is that correct?
We do have some funds in the core.
There are some funds for PUI campuses in the core to invite select people based on the need to come here over a 2-day period to learn and perform a specific experiment. Yes.
Thank you.
And we'll be after we get done with the 3 lectures, we'll advertise that and we'll invite people based on need and based on matching their interests to come through this training session.
Thank you.
Welcome. Okay. Anybody from here, guys? I see all these guys all the time, so they can ask me questions.
That's why I asked them first.
Yeah. All right. Thank you very much. Don't be afraid to ask. Send me an email.